Well, as you see in the screen, my name is Ana Ruescas, Ana Belén Ruescas. I'm from Spain. I work as associate professor at the University of Valencia in the geography department. I'm a geographer. That's the reason I get lost every time I visit a new city. And I also help Karsten Brockmann, which is the CEO of the company, as an external consultant with some issues like this one. When he has a meeting, I can come and give you this talk today. And I'm part of, have any of you ever heard about Snap software? Okay, so I'm part of the team and I help with the quality tests and also in the STEP forum that I hope you know, and if not, I will mention it during the presentation. So I work in water quality since 2011. When I joined Brockmann-Consult, I spent almost 6 years in Germany working with them before I moved back to Spain. And nowadays I'm more used to give lectures at the university about hydrology and biogeography, but this is also part of my background. So I wanted to start this presentation by reflecting a bit, giving some thoughts to the water quality and linked it to human health. So we need good ocean and fresh water because they are synonyms of health environment, of balanced ecosystems, and of, as a consequence, high quality food, which is very important for us, no? Especially in all food chains, but especially for us. And I put there paella, that is a typical plate from my city in Valencia, in Spain, that is Valencia. And when there is a lack of good water quality due to natural or more often anthropic causes, this balance in the ecosystem breaks, and that can affect our health. For instance, if we produce eutrophication of water, that means if we throw to the water more nutrients that they can absorb somehow, no, they convert into proteins. Some of these cases of phytoplankton blooms can appear in the water. That happens, for instance, in lakes that are surrounded by cultivated areas, and there is heavy rain and runoff, and a lot of nutrients arrive to the lake, and then the quality of my water is going to decrease because it's going to be full of microalgae that can affect also the life in the water. So certain species of this bloom, the algae blooms or phytoplankton blooms, as we call it, have powerful biotoxins, and they are, for instance, the cause of the red tides. I guess you have heard about that. And these toxic blooms can kill marine life and people who eat this food, no, this seafood. Phytoplankton can cause mass mortality with causing this deoxygenation of the water and causing this what is called dead zones. And it can also be aerolized. That means transport into the air and arrive to the coast, and we can breathe it. And these are all toxins that are in the air, and they can affect also our health. So we are going to focus today on explaining how we can derive some water quality indicators from the remote sensing sensors. So there you have some indicators that can be extracted, can be quantified using satellite sensors. I put some of the most famous, like the diffuse attenuation coefficient, turbidity, the water color. We will see what is this water color later, the water clarity, suspended sediment load, and euphotic zone, how far the light can reach in the water. So as you see, all of them are related somehow to the transparency of the water. So how far the light can reach in the water and what kind of response or spectral response we are going to obtain. In the pictures that you have there, you have like the water clarity is related to in situ measurements taken with a Secchi depth. That's a disk that is introduced in the water and is measured the depth at which you cannot see it anymore, it disappears. So it's so there are many of these indicators that are linked to in situ measurements, because traditionally we have measured our water quality in situ, not with satellites. Traditionally, I mean in the last 200 years. Okay. So these indicators can be ecological indicators. They are quantitative measurements or measures of the ecosystem and are typically based on the presence of phytoplankton. And from space, we are able to quantify this phytoplankton. And we have a series of indicators there or products that we can derive by using remote sensing, like the phytoplankton biomass, the primary production, that is the chemical energy that are produced by living organisms, so the translation of radiation light into proteins, basically. Phytoplankton size and community structure, and this is important, and we'll see later why. And also, we can study the phenology of the algae that we have in the water, of the blooms. So why phytoplankton? Phytoplankton are microalgae. So they are similar to terrestrial plants, and they are based on the same. They need light to, with the photosynthesis, produce nutrients. So they have chlorophyll A and other types of pigments. And this chlorophyll A is the parameter we can measure with optical remote sensing, optical sensors. Okay. Most phytoplankton are buoyant, and you can find it near the surface of the sea, in the upper part of the ocean at least. And when sunlight penetrates the water, we are able with remote sensing to measure the response of this algae. Phytoplankton also has some inorganic substances and matter in between, and especially because it needs of these inorganic nutrients like nitrates and phosphates and sulfur, etc., to grow. Phytoplankton, I mean, remote sensing ocean color scientists are obsessed with phytoplankton because it's easy to find. They live everywhere. They live in every aquatic environment. They are an integral part of the Earth's ecosystems because they are the base of the marine food chain. And they also are part of the biogeochemical cycles, and they affect and they are affected by climate processes. So as I told you, traditionally, phytoplankton has been measured in situ, first by observation, then by collecting bottles of water and then analyzing them in the lab. There are other types of in situ measurements, like sensors located in buoys or in robots that go down from the surface to the deep, like the biogeochemical Argo floats. And but long-term and large-scale biochemical dynamics in many marine ecosystems are still very poorly if we only trust the data that is provided by these in situ measurements. So we need the satellites to enlarge the spatial and the temporal scales of this type of measurements. As I told you, phytoplankton from space is measured by a proxy that is the chlorophyll A content in the algae that form this phytoplankton. Okay. So these measurements with satellite sensors of the chlorophyll A has been taken in the last probably more than 30 years. So we have a large series of temporal time series of chlorophyll A from space from different sensors. And we are, from this chlorophyll A, we calculate this phytoplankton biomass, and we can also derive parameters or indicators like the primary production. And we can link these dynamics of the primary production and changes in primary production to multi-decadal changes in the changes in the physical ocean, like the oscillation and of the Atlantic and the decadal oscillation of the Pacific. And I also mentioned that these phytoplankton sizes or communities, and this is also an important issue because we have, maybe I can go back to one previous slide just to show you. We have, there are thousands of algae species. There are many of these, but we can group them by size in 3 groups, like the pico, the nano, and the micro. So pico is below 2 micrometers, nano below 20 micrometers, and micro below 200 micrometers. So we can measure the abundance of this phytoplankton, and we can also determine with models that calculate the fractional contribution of each size of plankton. What is the main size on the different parts of the globe? So we in this image here, I know the literature is a bit old. Everything goes so fast right now that is almost impossible to follow everything. But I like this image because it's showing very clearly that there is for instance, we have very fertile waters in the high latitudes. The microplankton, this one that is less than 200, like the biggest one, let's to say, lives in these waters that are seasonal because part of the year they are also frozen. So in spring, when they start defrozen, there is a big mix of water and a lot of nutrients that are in the bottom of the seas arrive to the surface, and they take advantage of the light in the upper part of the ocean. And these microplankton are the most nutritious and the most important in our oceans. So if we see, for instance, the distribution of the phytoplankton, the smallest one, we see that the abundance is higher in areas on the subtropical gyres, usually where the ocean waters are more stable. There is less stratification. And these waters are less, they are not as rich in species like the high latitudes waters. So what happens with temperature increases and climate change, that this distribution of types of sizes of plankton will change. And that means that our waters will become less fertile. Okay? We will have more phytoplankton or nanoplankton in higher latitudes with this global warming. This is one of the studies and conclusions that we could extract by using chlorophyll A from remote sensing. We can also use this information, for instance, to understand or to help fisheries and the aquaculture industries because phytoplankton are a source of essential fatty acids. And these fatty acids are essential for the survival of other species, especially for vertebrates. And the different types of phytoplankton produce also slightly different fatty acids. So we can help to map these fatty acids by using chlorophyll A as a proxy too. We can do more with remote sensing for fisheries and aquaculture industries. For instance, these HABs, these harmful algal blooms that I mentioned in the first slide, can be detected because they are usually related to changes physical changes in the water. So there's a higher rise of temperatures usually, and some other indicators. So this detection and warning is possible using remote sensing. As a consequence, we can determine if there will be some hypoxic events depending on the abundance of the bloom and the quantities that we find. And we can also, other uses could be to select the best place to locate some of our farms, some of the places where we will establish these cultures of mussels or other aquaculture species. I also wanted to talk a bit about freshwaters, how water quality is important also for urban waters. And we saw yesterday in the museum that they are now trying to make the people drink more tap water. And I have to say, I'm going to support this idea. This is something that we have in our houses, and we are so privileged to have this. I mean, this is amazing that we have water from a tap and we can drink it directly because it's good quality water. It's controlled by the authorities, so please do it. So for recreation uses, it's also recommended too to have good quality water. And Brockmann-Consult had this project in Hamburg to monitor the lake and the river Elbe water quality. And I will show you some examples now. So this is more or less the idea. This is a chlorophyll content of the Elbe River in Hamburg. And there is a service that allows, I mean, it's a downstream service, allows users that don't have any experience with remote sensing to use remote sensing to establish when the quality of the water is good or bad, and give alerts, for instance, for bathing waters. When it's better not to go into the water to avoid intoxication by algal blooms, for instance. And this service is provided daily. And I also wanted to mention, just as a closing introduction, there are other hazards related to water, and I know you have seen some of them already in previous presentations. So how we can work with remote sensing with satellites to, for instance, in this case in Mozambique in 2019, the path of the cyclone Idai was devastating, and it affected 3 countries, Malawi, Zimbabwe, and Mozambique. And all that area in red is the area that was affected by floods in this case, and 1 million of people displaced. And we have the recent case of the Libya, and how remote sensing can help to, not to avoid, well, so we should use it to avoid or try to avoid these disasters, but when the disaster has been, is produced, at least to do an emergency cartography, emergency mapping, and so the rescue teams know where to go exactly to rescue people if possible. This case in Libya, for instance, cause this big plume in the ocean, in the coastal ocean waters. So all these sediments dragged by the water arrive into the sea, and also cause a change in the color of the sea and in the components of the waters of the coast. This is one of those plumes. And that, of course, affects the ecosystems that are there. So we also see this plume in a lake, in this case, in Israel, that is also caused by intense runoff and drag of sediments. So I found this slide of water-related disasters in this epoch from 2001 to 2018. And yeah, it's noticeable, I think, to both floods and droughts, to see how probably the biggest countries with highly density population are, of course, the most affected. Maybe not in the case of the United States, but if we go to Asia, China, or India, we see here that in total deaths and affected people is really, really high. So in the following slide, I have these health impacts of water-related disasters. This is not the cases in itself, but what floods or what droughts can cause in the health later. And for instance, due to floods in 2020, almost 240 million people were affected by malaria. Around 4 million people were affected by cholera. And some of them not. Luckily, we have medicines. No, we can avoid most of the deaths, but still the rate is quite high. And globally, a lot of people still suffer from bad quality in drinking water that produces also a lot of illnesses. Water contaminated by feces. There are 2 billion people in the planet that drink water contaminated by feces. So this provision of safe drinking water is compulsory, is important, and we are so lucky to have it. This is my message. Lately I've seen there are a few works that are try to understand better, for instance, when these cholera outbreaks are going to take place using remote sensing. Is still difficult, and there is not a direct link in between phytoplankton and cholera, but well, some people are trying to find this link via indirect products. For instance, once we have a phytoplankton in one area, the following the other people, no, sorry, the zooplankton will follow and will eat this phytoplankton. And there is a delay in this, like 1-2 months. And with the zooplankton, a lot of bacteria of the Vibrio cholera arrives too. So maybe there is one way of saying maybe we could have an outbreak now when we have a strong phytoplankton bloom near some inhabited areas. There are other researchers that they say there is a direct link, because there are some biofilms that are formed of this bacteria around the phytoplankton. But it's a work that I think is still not mature enough, but is in a good path. So I've been talking about phytoplankton, and how we measure phytoplankton from space is still not very clear, I guess. So to measure phytoplankton, I told you what we do is to measure or to observe what is the water color. And this is the basis of the water quality and the measure of chlorophyll A and the phytoplankton. So what is the color of the water? We have here several waters with different colors. So what this is showing us is that our waters have different contents in the different matters in there. When we have this type of blue colors, they are usually related to clear waters. When we have these green colors, or this one here, we can link these colors to some chlorophyll content. That means there are some algae in my water. When we have this brownish, it could be the decay of the algae, or it could be sediments in my water. And sometimes I have like very dark waters that are caused by color dissolved organic matter. I tell you what this is a bit later. That absorbs practically all light. It's really dark waters. So color of the water is just as it sounds. It's sunlight interacts with the ocean. Some sunlight is reflected back by the surface of the ocean or by the waves, etc. And we are not really interested in that part. What we want to measure is the quantity of light that is entering the water, interacting with the matter that is there, and then going back. Okay. We know that by definition, water has a blue color. So that means that it's this part of the spectrum that is reflecting or is arriving farther away in the deep and is reflecting more when we only have water molecules. But we will see when we have, for instance, some algae in my water, my spectrum will change. So this is a blue water spectrum with reflection in the blue part of the spectrum, 400 until 450. And when I have water, when I have algae in my water, it becomes green water. No? The peak of the spectrum moves to the green part. No? It goes to longer wavelengths. So I was telling you, what I'm interested in is not this surface reflected, but is this water leaving radiance, or what is leaving the water after the interaction with the matter, organic or inorganic, that they are here. So there are 4 processes that can be, physical processes that can be taking place inside the water and interact with the matter I have there. So most of the light can be absorbed, or it can be scattered. There is a process called backscattering, that is what we measure with remote sensing, and also the fluorescence that we don't see it today. Okay, but this is an important matter too, because there is a satellite that is going to fly soon to measure fluorescence inland, and we hope that also in the water. So this reflectance, this water-leaving reflectance that we are interested in is this one, and we know is a composition, a composite of all the absorption properties and the scattering properties of the matter I have in the water. Okay. So the main substances that affect the water color are the phytoplankton, as we have seen, no? With chlorophyll A pigments, resuspending sediment, so all the sediments, usually non-algal particles, so mineral particles that arrive to the water, for instance, from river with river plumes that we have seen before, they don't absorb as much as they scatter, so they reflect a lot of the light, especially beyond the red or yellow and red part of the spectrum. Just to make it clearer, so the spectrum of the water is this one. We see the low values of the reflectance that we have, and I will explain later why. Okay. This is the blue water, let's say. Okay. When I have sediment in the water, my signal is going to increase, but it's moving to longer wavelengths, no? To yellow, brownish colors. When I have algae in my water, the peak will show in the green part of the spectrum because it's an effect of the chlorophyll A pigment, and also on the near infrared. There is another peak here, okay? And this is a signal of chlorophyll water. And then I have this color dissolved organic matter. This color dissolved organic matter is, it could be produced by decaying phytoplankton. So when the phytoplankton starts to decompose, or also by the residues of plants that grow close to my water mass that arrive to the water. This is producing this decaying matter, organic and color, that absorbs practically all light. It only reflects a bit, you know, in the blue, but it absorbs practically in the rest of the spectrum. And with this basis, I can understand what I have in the water, you know, depending on the, yes?
The water that has like microplastics and so on, is it also changing the spectral signature?
It's also
Has microplastic contamination?
microplastic, there are a lot of, well, new research going in that direction. I haven't shown anything here, but now they are trying to understand how it affects the signal too. But I cannot tell you right now. But I know there are a few works ongoing. So I told you that the two processes that are more important for watercolor remote sensing are absorption and backscatter. And these are known as the inherent optical properties. So these properties depend only on the composition of the water. It doesn't depend on the light that arrives. And this is why they are called inherent in this case. They have an additive property. That means in a multi-component medium, the total absorption or the total scattering is a sum of the different or individual contributions of all the matter that we can find in the water. By contrast, or besides, we have the apparent optical properties that depend on the illumination conditions, depend on the solar zenith and azimuth angles, and they are I think we all know, irradiance, radiance, reflectance, all these variables related to the interaction of light with the surfaces. I wanted to give, I think I have enough time, only just for you to understand, because I think comparing with land, the land signal, the signal is very different. You know, the land, you have this high reflectance on the beyond the green and on the red and near infrared, and here we have these funny shapes, and I wanted to spend a bit more of time on this, if you don't mind. So we said that in the case of the phytoplankton can absorb, but it can also scatter. But mainly what it does is, the characteristic that we measure is the absorption of the phytoplankton. And we have a specific absorption coefficient of phytoplankton, as I told you, that with a peak this is the sum of all the pigments, let's say the chlorophyll A, that moves to the green, and another peak of absorption that moves to the near infrared. So this is the chlorophyll A, but there are other pigments in my algae. There are chlorophyll Bs, chlorophyll Cs, and other particles. But what the optical sensors measure right now is chlorophyll A. And this chlorophyll A is our proxy for phytoplankton. Okay. This signal varies with the species. So depending on what type of algae I have, of microalgae I have, the signal varies a little bit. but this is also very useful to classify in species when possible, and to produce those maps that we have seen before, at least of phytoplankton sizes or phytoplankton communities. Okay. So the scattering is calculated in phytoplankton, is calculated as an exponential function that depends on the wavelength and the content of chlorophyll A. So this is more or less what it looks like, and this is a formulation done a long time ago. I think one number is missing here. So for the sediments or non-algal particles that I have in the water, we could maybe, we have suspended material that can have an organic nature or an inorganic nature in this case. The absorption is usually very high in the blue and decreases exponentially to the longer wavelengths. And the backscatter, as you see, is quite high, practically from the green band till the near infrared. So we for optical water quality, we focus mainly on until the 800 nanometers to the 400, 800 nanometers, more or less. The backscatter is usually dominant in inorganic particles and in coastal waters or estuaries or rivers, etc. So in open ocean, we don't usually have much of these other components beyond the phytoplankton. So, and then for the color dissolved organic matter, we have. It does probably, well, they don't scatter, they absorb practically everything. They have this peak of absorption in the blue, and but they are a complex mix of humic and fluvic acids, and usually resulting from the terrestrial vegetation, or as I told you, is a decay of the phytoplankton cells and other organic plant particles. And sedum is very important for high latitude lakes, for instance, in Sweden or in Finland. In Finland particularly, I don't know if there is any Finnish in the room, but half of the country is shallow lakes, and they have this dominant sedum lakes, very dark, and they absorb practically all light. So to study sedum, usually there is a correlation also with chlorophyll A and salinity, and sedum can also suffer, like other organic matter, photodegradation. So there is also a nice research field for sedum researchers. So how do we use this IOPs, this absorption and this scattering, to understand how do I use my signal, my reflectance measured by my sensor, to extract these substances? What I really want to know is I want to quantify how much chlorophyll A I have in the water, how much sedum I have in the water, and how much sediments I have in my water. This is what I want to know. So we can use a direct model to go from these substances measured in situ, and then I measure the absorption and the scattering properties of each of these variables, and then I relate it to reflectances, or I can use an inverse model. So that's what we do when we use remote sensing. We have a satellite image, we have the reflectance, and we want to extract the concentrations. So we usually do it using very complicated bioptical models. And so if I want to plan to do that in a batch mode, I will need, so this is one example. I took it from a NASA training. So I have a satellite TOA reflectance or I have a satellite BOA reflectance over a water body, and I have some in situ observations of the water quality parameters I want to derive. So what I do first is my satellite TOA, I apply an atmospheric correction to extract the BOA. In this case, my BOA is my water leaving radiance, is what is my interest, not the reflectance as we saw. And then I do a statistical or empirical adjustment between my in situ and my reflectance and extract an empirical algorithm. So I extract some model coefficients that I can apply to the rest of my images. No? This could be a way of going. So the atmospheric correction in water is very, very important, and is, I would say, I know for land is also very important, but in optical sensor, atmospheric effect on the water signal is very, very high. So this is, if we look only, don't look to this part of the, so if we look only until the 800 nanometers spectrum, what I have here is that from the signal that I receive in the sensor, 0.9 or 90% is due to the effect of the atmosphere. Of this, a big part is due to the Rayleigh scattering, and I can correct for the Rayleigh scattering using some illumination geometries. And then I have also a big contribution, especially moving to longer wavelengths from the blue to the red near infrared, of the aerosols. And these aerosols are the difficult part to correct, and we all know that, okay? So the red line is this surface reflectance that I don't care about. What I care is this signal here. So in water, for the blue part of the spectrum, I have a maximum of 10% of the signal that arrives to the sensor. So I need to correct my images and to remove all these effects to get this. And this makes really this part of the processing very, very important. And if it's not well done, it can cause a lot of problems. It can give me wrong values of concentrations, etc. Once I have this water leaving radiance or water leaving reflectance that I'm going to use, I can apply simple algorithms to derive chlorophyll A. So this is the OC4. This is one of the most used algorithms. It's the one that is used by NASA, and it's the one that is used by EUMETSAT or an ESA to derive chlorophyll A. And what it does is just to compare, or it takes the maximum of 3 possible ratios with some in situ measurements of chlorophyll and extracts the relationship, is what I showed you before. There are other ways to doing this, by inversion models, using also semi-analytical relationships. So using not only this direct relation with the concentrations, but using the absorption and the scattering parameters, or or even we can go beyond in using some machine learning or deep learning models like neural nets. So one of the products that EUMETSAT is also providing with the level 2 data that I will tell you a bit later is the chlorophyll derived using a neural net with an algorithm called case2 RCC, case2 regional ghost color, that Brockmann-Consult developed with other partners a few years ago, and it's still in use. So you have 2 different products of chlorophyll and sediments you can compare. Usually, as I said before, this OC4 algorithm is designed for blue waters, for waters where open waters, where my only contribution to change in color is the phytoplankton. But if I go to coastal or lake inland waters, I will have more things in my water, and this algorithm, this relation is not going to work properly. So that's why we need to apply other methods like neural nets or other machine learning algorithms to derive this, or try to find a better correlation. So this was a video. It will take a while. So what we have right now, as I told you, we have measurements of the ocean color products from the last 30 years. Probably NASA started with some of the first sensors on board of sea waves, and then MODIS. I think there was another one before. I don't remember the name. I will talk about it later. This is an animation I created with the Ocean Color Climate Change Initiative data it's a program from ESA. And what we can now do is this type of level 4 products, global ocean chlorophyll data, that we can use together with other variables like sea surface temperature or sea surface height, so altimetry data on the level of the waters. Okay, this is what I wanted to show too, how these 3 variables can work together to do more precise ecosystem monitoring. So from, if we combine ocean color with sea surface temperature, we can grow our field of research with some topics I have already talked about, like harmful algal blooms, HABs to shorten it, plume dynamics, coral bleaching, and all related to human health. When we mix it with sea surface height, we can also do some research on sea ice dynamics, underwater volcanic eruptions, internal waves, transport of blooms or transport of pollutants. And if we mix sea surface with sea surface height, we can also research on eddy dynamics, anomaly, marine safety, forecasting, and storm dynamics. Okay, as in every other topic, so land topics, land-related remote sensing research, we cannot have everything. You cannot have everything. So we always have to decide from everything we have available, all the programs we have available, what is the one that fits better my target, my research. So in this graph here, what we have is a graph comparing the temporal and the spatial scales of the actual remote sensing ocean color satellites. And what we see is that usually there is a trade-off in between the time and the spatial scales. This is, I don't think I have to go really farther into this, because you know already, because in land it's, I guess, land is sometimes a bit less dynamic, but the problem is practically the same. So yes, to mention some of the satellites that are used for ocean color. So SeaWaves, MODIS, VIRS from NASA, Sentinel-3 from ESA, Sentinel-2 and Sentinel-1 from ESA too. So we have the Landsats from NASA, Prisma, there are third-party missions, I think. like some other hyperspectral map, for instance. And we'll see later what this L4 and L3 means in another plot. So you have the spatial scales here. No, if we go from very, very particular site scales to local to regional, etc. So this is the swarm of satellites that are right now observing the ocean. So there are, as you see, many of them measuring not only ocean color in this case, but other variables like ice or temperatures or heights. And I wanted to start with showing you some examples of the NASA products, and then we'll go later to the ESA ones that are the most important for us. But yes, to show you there is really a long time series of ocean color data. So we have the most important sensor for ocean color research is right now from NASA is MODIS. Also VIRS is a continuation. And it covers the Earth's surface every 2 days. It has 36 bands at 300-meter pixel resolution. Okay. It was launched in 2002, and there were 2 satellites carrying this sensor, Terra and Aqua, and it's still ongoing. So there is 20 years of data, but it's soon coming to an end. This is one of the series that is going to finish. We will have bears after this, and we had seaweeds before, so there is a long time series of data. I wanted to mention that NASA has this ocean color web. So this is the Ocean Biology Processing group, and they have a web where they store all information, and there are a lot of not only where to find the data, but also some resources for training, for instance, tools that are used to process this data, a gallery, and a community, a forum, etc. So in next year, early next year, they will launch also the PACE satellite, and this is important for ocean color scientists because it carries a hyperspectral sensor that is called the Ocean Color Instrument, OC, very similar to OLCI. That is the one we use in Sentinel-3. And don't remember exactly the characteristics of the hyperspectral right now, but it's medium spatial scale, but it has more than, I think, I don't want to say any lie, but so for instance, the hyperspectral we are using now in Europe, that is Prisma or EMAP, they carry around 200 bands, so it must be around that. It carries another 2 sensors, an altimetry sensor, I think, and another one that sees in different. It's a polarimetric sensor. So the idea is to work with all of them together. In Europe, we have the Copernicus program. The Copernicus program is a European Commission fund, and all the Earth observation data are freely available to any users. You probably are more familiar with the Copernicus Land service, or also with other services like the atmosphere, or emergency, etc. They are dedicated to satellite operation and data services. And in the case of the marine program, the Copernicus Marine Service is shared between ESA and EUMETSAT. And I will show you later too how. So I don't think I need to spend time in this slide. I guess you already, do you know the Copernicus program? Yes, okay. So this is one of the last slides I managed to get from them. I'm pretty sure you have seen this slide before too. So this is all the satellites that have been built and launched by ESA. So we have these three main orbits, no? Like the science one, the Copernicus, and the meteorology. The meteorology is usually these Meteosat satellites for weather forecasting. Then we have the science that are like satellites that are more limited. They have a lifetime that is a bit more limited, and they are launched as a proof, like to test if the science behind the sensor is going to work, and then to make them operational or to convert them into sentinels at some point. And then we have the sentinels that are the operational satellites that the Copernicus program uses. And in the case of ocean color, or in the case of water quality in general, we use both Sentinel-1, Sentinel-2, and Sentinel-3. Now Sentinel-6 also carries an altimetry, and they are all in place, and they are also, I think next year, maybe Rafael knows better, they will launch new Sentinels-3C, or in this case. So we use mainly for ocean color Sentinel-3, Sentinel-2, and then for altimetry Sentinel-3 and Sentinel-6. And for temperature, we use Sentinel-3. There are different sensors in the same satellites. And there are other interesting satellites here. Time is hyperspectral too. And LSTM is a thermal high-resolution sensor too. So the future is very promising, I think, for data and new capabilities that have to be developed. So Sentinel-3 is is what is called the blue satellite, because it's the one that has more sensors dedicated to ocean on the Sentinel series. And it carries these three sensors: the SLSTR that measures the temperature, the ocean and land cover instrument, the chlorophyll that measures the chlorophyll, and the altimetry that measures the height. For the marine part, EUMETSAT operates the Sentinel-3. Also, ESA operates the Sentinel-3 for the land part. Okay, and the Copernicus Marine Data Service provides all L3 and L4 products from the Copernicus, or from the Sentinel-3. So some examples of what OLTSI can do. So OLTSI has a full resolution of 300 meters. So it measures in 300 meters, and then there is another product in full resolution, in reduced resolution, 1.2 kilometers. It has 21 spectral bands, visible and SWIR. It has an excellent signal-to-noise ratio. This is important because we will see later that there are other sensors that have higher spatial resolution, but the signal-to-noise ratio is lower. So sometimes it's good to merge, but not to only work with one of them. And there is something different in my presentation. It doesn't matter. So everything about how to process the data is all available in the internet, user guides, handbooks, etc. There is an S3BT validation, S3 validation team, Ocean Color meeting at the end of the year. Also, I think also people from land and thermal, they are also part of it in Darmstadt in December. For Ocean Color, we also use Sentinel-2. Sentinel-2 it was designed for land studies, but it also takes part of the coast and inland waters. So we can apply some of the algorithms we use for OLT, they are adapted to the bands of the MSI, multispectral instrument. It has a maximum resolution or maximum pixel size of 10 meters, but also have different bands in different pixel sizes. And yeah, it's ESA who is managing the level 1 and level 2 data of Sentinel-2. So some examples, this for coastal and inland waters, we have here the results of these sediment plumes in the Florida floods on 2022. Don't know what else I can say about this. So observing surface blooms, monitoring water quality, monitoring aquaculture farms. It has capabilities with these 10 meters to map some of these aquaculture farms. It's also used for validating other applications with radar, for instance, or detecting floating debris, no? The plastic that we were talking before. This is an example on the Sentinel-2 performance on a lake in Sweden. And as you can see, this is the water-leaving radiance that we have after atmospheric correction. And this is the chlorophyll product that we obtain once we have these reflectances. So as you can see, we have here 3 different types of water. So one that is like green, no? Green water, that it results on very low values of chlorophyll, but still some. This greener water with some parts affected also probably by sediments. And then we have this case of these dark waters. These dark waters are the product of a lot of dissolved organic matter in it. And the algorithm that we have right now designed to extract chlorophyll, in this case is the neural net, is not really trained for these types of water so far. So it's still difficult to find a good algorithm on Sentinel-2 for very dark waters. So this is one of the areas of study that is still open. So since I only have like 25 minutes, I don't know if we need to go through all this. This is just a comparison of just a reflection that it depends on what you want to obtain, you will need a type of sensor or another. So it's true that with higher spatial resolution, we have more detail, but we can also have more problems. For instance, in the case of the Sentinel-2, 10 meters resolution, we have a very nice view of the coast, but we also have this sangling problem. So we have an angle of view of the sensor that is not optimized for water for observing waters. No, Sentinel-3 OT, it is optimized for this problem, but not Sentinel-2. And then we have this very high resolution commercial satellites. We have 3.6 meters, but the radiometric resolution, the signal-to-noise ratio, is not as good. So we have to compensate a lot or to think what we want to obtain and what we need, and then to use the proper sensor. So this was a comparison of the spatial versus, well, this is more the spectral, in this case, comparison between Sentinel-3 OT, the 21 bands and the 13 bands of the Sentinel-2, and how the width of the bands is also different. In Sentinel-2, we also have to take into account the different spatial resolution of the pixel size. And in this arrow, we say the signal-to-noise ratio is lower for Sentinel-2 compared to Sentinel-3, so the quality of the data is going to be, or the precision of the data is going to be a bit lower. I wanted also to compare Sentinel-2 with some commercial satellites, because lately these commercial satellites are improving a lot. So before we only had these 4-band high spatial resolution satellites, and now they are adding more bands. They are doing the bands narrower, so they are even comparable with some of the operational satellites. So still we have to pay for them. So this is another thing to have into consideration when working on those topics. Yeah, and this is just an example of we have more detail on the dynamics of a bloom, for instance, when we have a higher spatial resolution, but because of the spectral and the sensitivity of the sensors, maybe we are not able to, with this type of data, to obtain all the signals for the different types of algae that I have in my water. So this is something we have to consider. Okay. So since I don't have much time left, I had two other parts in the presentation. One is about where to get the data, and the other part is about how to process the data. Well, which tools I have to process the data, and we have to choose. So, what? Then? Should I continue until I finish my time? Or? Yeah? Okay. Well, I think that the PDF of the presentation will be given to you, and I will be here until tomorrow. So if you have any questions. Okay. So what data are relevant to us? So as I told you, after all this introduction to the ocean color, so what we need to think what variables we want. And sometimes we want variables that we think we want, but we realize later that we don't want that. We want another thing. That's why we think we want. What area, one time period we want to cover. And but also we have to take into consideration other things. So the main problem that we have now is that we have such a quantity of data, so a massive quantity of data, that every time is harder and harder to work locally, for instance. And besides, data is usually located in very different programs and services. It's distributed all along, and we need to understand where to find this data. And then this acquisition and then load is time consuming. So in our project, we have to also always have some time to do this task, or at least to think how we are going to do it. If we are going to work on a cloud, or we are going to use some servers, or if we are working locally. There are so many additional considerations we have to take into account. Spatial coverage, spatial resolution, spectral resolution we talk about. So the standard algorithms that are in the market are suitable for my area. So if I apply an OC4, it's going to work in my lake nearby. The temporal revisit. Is a time average suitable? Do I need a specific date, a specific time? Why do I want a better quality? I think I can improve the quality. How much data is lost? So the flagging is a very important issue when working with remote sensing, and I know because I'm the first one that does it. We don't use it. We don't use it as we should do it. I mean, take into consideration of these flags that are produced by ESA and EUMETSAT when they process or reprocess data. Do I need one single sensor? It is better to work with a merge, with a synergy of sensors. The format, what is the easiest format? Is the data free, or do I need to buy it? All these things are things we have to know. So I put here what I took from Mem Loveday. So I'm working with him. I hope he doesn't mind. This table with the processing levels, I think these processing levels must be really similar to the land levels. So we have this level 0 that is usually not distributed. The ESA and EUMETSAT are keeping. There's the data that are receiving from the sensors. Level 1, it says here A plus B, but I would say also C, for instance. Sentinel-2 data delivers also level 1C, TOA reflectance data, not TOA radiance. So I would like to add this C here. Reconstructed, unprocessed, top atmosphere instrument, data at full resolution, and annotated with ancillary information. Level 2 is already derived geophysical variables, same resolution as level 1, but is usually atmospherically corrected, but we need to check if the atmosphere correction is good enough. Level 3 variables map on a uniform space time grid. That means that usually I don't have only one or the data from one sensor, but I can have a synergy with other sensors or merging with other sensors. In the case of ocean color, I will merge OLCI with MODIS, with BIRS, with SeaWives, and have this level 3 product. And then level 4, that is the model output or results from analysis, the downstream products that deliver, for instance, the Copernicus services. Are already gap fill, temporally aggregated, daily, global data. Okay. So in this plot here, we have also a graph of convenience versus flexibility. So this convenience means how easy is the data for you to use. So we have that these third-party web GIS products that deliver images are the most easier for us to use, versus the L10, that is the data that is less convenient for me to use because I don't understand this matrix of number. So this is talking about, let's say, the level of processing that has been applied and how easy is the data to understand. So we usually as scientists start from level 1, but we can also do a lot of work and nice science using level 3 and level 4 data that are delivered by, for instance, the Climate Change Initiative or the Copernicus Marine Service in the case of ocean color. So satellite providers and downstream services. Level 1 and level 2 is more for the basic science, to improve the algorithms or to build better processing chains. And these are data delivered by or provided by satellite operators and data providers, like EUMETSAT, ESA, NASA, NOAA. Flexibility is against the convenience because, of course, one image, as I told you, is already there. You cannot really change any parameter. So the flexibility is higher when the data is rawer. when there is less processing, you are more capable of changing what you have. So data providers, you probably know all of them. ESA and EUMETSAT for the marine service of the Sentinels, CNES, DLR, NASA, NOAA, and other special agencies, the Korean, Japanese, etc. Also some private companies or private services, like Planet or ISI or Digital Globe that are now providing commercial satellite and high-resolution satellite data. About the programs, well, Copernicus is the focus of our interest here, but also we have the Climate Change Initiative. Inside Copernicus, we have this Copernicus Marine Service or Copernicus Land Service that delivers the inland waters services in our case. The CARES for the SST, the data from satellite and models for the ECMWF. And some of these dias that I always have to note what it means, because I always forget. So these data information and access services, like Wikio or CreoDias or others. Also some private solutions, some cloud services are here. The Sentinel Hub that I guess some of you use too, and etc. And the SAF, the OSI-SAF. So the Copernicus program, I guess you know already. In the case of the marine data, you see here there is inside the program, there is a space section, a service section, and an in situ section. So the space section that is technically coordinated by ESA, the Sentinel missions are operated by ESA and EUMETSAT and other contributing missions, and it usually provides and delivers level 1 and level 2 data. If we move to the services, all the Copernicus services are fed by the level 1, level 2 data to produce level 3 and level 4 products. So you know the Copernicus Land Service, Copernicus Marine Service, atmosphere, or emergency, security, etc. And then we have the in situ part that is also important, that is participated by the different states that are part of the program and coordinated by the European Environmental Agency. So data repositories, we have, there are many more, I'm pretty sure. I put this as the most common repositories for us, from EUMETSAT with the data store that you can access by web interface or by APIs. The data center, that is an offline service. EUMETCAS is, you plant the disk in your apartment and you have Meteosat satellites in your house deliver every 15 minutes. EUMETSAT View is a web GIS. So ESA, before they had the Copernicus Open Access Hub, and it will be discontinued by the end of this month. And the Copernicus Data Space Ecosystems will replace it. And it's also a GUI and an API. Then we have data from the CCI portals. And there is an Ocean Virtual Laboratory that is also a web GIS service. I think there are conversations for cloud data services that are still ongoing. And then we have repositories from, as I told you, level 3, level 4 data in the Copernicus Marine System, also in form of GUI or APIs. And then there is the My Ocean Viewer. The DIAs represented here by Wikio, because Wikio is the one that EUMETSAT is more linked to, that's the reason. Also with GUIs and APIs and cloud services, so far as I understand, most of it has to be paid. There is a quota you have. And then NASA and NOAA that we are not going to go into detail. EUMETSAT OSI-SAF, for instance, if you want data about sea ice, you have to go to the OSI-SAF. They have various ways of accessing this data, APIs, FTPs, the ECMWF, and the Sentinel Hub the web browser that I mentioned before. So here there is a table that summarizes for the different topics concerning marine services, ocean color, sea surface temperature, surface topography, that is altimetry data, radar, and ice, the different levels that you can find, and the different service providers. This is not all that we have, but this is the ones that are more accessible for us, and they offer free data etc. So some things about this data shopping. Practically, I would point out two things in this slide. Is that we have to have in mind that the formats of the data are starting to change. We are quite used to work with NetCDF. Most of the Copernicus services and even the Sentinel safe format is NetCDF based. But we see that more data is delivering in cloud optimized GOT for SAR in the near future, and also the architecture of storage is starting to change to object storage, like the S3 buckets of new ways of catalog data, like Stack. So in this slide, there are two different ways of delivering the data from the services. In this case, the push services is related to this, for instance, the UMedcast. You have an antenna in your lab, and you are receiving this data. It doesn't matter. It comes every now and then when it have to come, and then you keep the one you want practically. And then the pool services, we had in the past all these different ways of getting this data, the DODS or the FTPs, offline archives, OpenDAP, HTTP, AirDAP, etc., WebGIS. And now we are in this scenario where we access data practically by HTTP and the OGC APIs, the WebGIS based. But due to the massive quantity of data we will have in the near future, we are moving more to cloud platforms. So we can start to forget about processing local. What is going to happen is we will have an API with our processing change, and we will send this command to the cloud, and this data will be processed there. And then, okay. So yeah, I don't think I need to explain this, right? Difference in between the GUI or the web UI and the API. So one is a machine to human, and the API is just a code that is pushed into the machine, and there is a connection machine to machine. And well, those are some examples of the products we can find in the Copernicus Marine service. And so we have this viewer that you can check what kind of data you have. There are so many different products produced by different data services, and that is overwhelming. We have SST, we have chlorophyll, we have CIs, we have, and we also have not only global products in different spatial resolutions, we have also regional products for the Mediterranean or the North Sea or the Baltic Sea in different spatial resolutions. So you need to understand very well what you want and what you need. This is very important. As I told you, water quality can also be fine in the Copernicus land service. So we have the lake water quality in 300 meters, and we have coastal water quality in a fringe of 50 kilometers along the coast in 100 meters right now in the Copernicus land service. How to process really, really fast. There are several, if we work in this high flexible, less convenient part of the plot I saw before, like with L1, L2 data, we usually want to perform this atmospheric correction and to extract water quality parameters. This can be done step by step. So first I do the atmospheric correction, then I extract the water quality parameters. We can also apply algorithms that couple both things, like S2RCC, and we can use several softwares on several ways of processing, like in the case of the NASA for processing all the American satellites and also the European, because they also have the processors. We use CDAS, the CDAS software. There are, I put here 2 links with videos. These are YouTube videos of lecture series that took place through the beginning of this year. It's hosted by the Trevor Platt Foundation and is focused on marine data. And they have 2 videos and 2 presentations about how to process water quality using Python scripts or using Julia, also as program languages. So also from the UMETSAT, there is a Copernicus Marine Service training, and they do at least 1 general course every year, and then specific courses every now and then, on not only on water quality, but also on sea surface temperature and altimetry. And if you just have to join the UMETSAT training space, and it's free courses, and you have to pass this process as you did here for the ESA courses. What I told you that I'm involved in this project in SNAP. SNAP is a Sentinel application platform. I'm not going through the presentation, but SNAP also has this capability of visualize the data. So if I want to study what I need, I would use this type of software before, like to see the data, what I can obtain in level 1, level 2. It also reads NetCDF, so I can also work with level 3 data. So it has tools for display the data, visualize, analyze. And in this case, this is a Sentinel-2 of the Thames River, I think, in London, and some sediment concentration parameters. And SNAP offers these tools for selecting a few pixels and displaying what is the spectrum of the few pixels that are located here. Water, sand, crops, they are displayed here. It also has a lot of tools for data analysis. So profile plots, statistic analysis, extraction of points through time series, comparisons of scatter plots and profile, no, I already said. Well, for comparison with, for instance, reference data matchups. And it also has many processors to pass from level 1 to level 2 data in OLTI and MSI is possible, for instance, through using the K2RCC algorithm that I mentioned before, this neural net. But it also reads the standard Sentinel-2 level 2 and Sentinel-3 level 2 data provided by ESA and UMETSAT. And it has the capability of doing batch processing. So if I have enough capability in my machine, in my local machine, to store 1 year, 2 years, 3 years of data, I can do batch processing using SNAP. There are several ways of doing it, graphically, or using a service called GPT, or using Python scripts. I can also access SNAP using the Snappy or the Snapista. There are several ways of doing it. I think we have to take a break. I have to. So, well, the rest of the presentation is there, and you can take a look. There is a lot of user support. There are tutorials, the user forum that is very useful for the different satellites, etc. There is a community. You can collaborate, help others, so etc. This is my information, my email at Brockmann-Consult, and if you need anything, just write me. And yeah, thank you.